That's weird. It's happening. This happened before, and when I go back to my office, it seems to have settled down. I don't know why, not quite sure why there's a problem once I put it up. I know when I checked things, it seemed to be working, which is a little odd. I apologize for the problems, and I'm not quite sure where they're coming from, because the last time it happened, I went back and checked, and everything was fine, and nobody had any problems after that. So I'm not saying there's not having problems, because one person showed me the problem, but that's just kind of odd. I will check and see what I can find out, so I apologize for that. We are moving into a new area, and that area being signaling. And signaling is one of the most fascinating things that multicellular organisms do. If you think about it, and you probably haven't thought about it a lot, if you think about it, that it's necessary in a multicellular organism that all of the cells of the organisms have to be working in conjunction with each other. All right? That doesn't mean they're all doing the same thing, because they're not. We will find, for example, that liver cells are really good at making glucose, and so they may be making glucose while muscle cells are using glucose. And so they don't have to all be doing the same thing, but they all have to be working in support of the organism's best interests. And so we'll see several ways that the body, when we talk about signaling, we're talking about the body, how the body manages things. The responses are managed at the cellular and tissue level, okay? But the signals are happening at the organismal level. Signals largely travel through the bloodstream. We'll see a couple of exceptions to that, but signals largely travel through the bloodstream. And signals largely are relatively slow. We'll talk briefly about the nervous system being a signaling system, and it most certainly is. But it's of a different kind of signaling than is happening in the signaling that's going through your bloodstream. The nervous system can respond in milliseconds, and things in your bloodstream can only happen as rapidly as the blood is moving through your body. Okay? So you remember those things, and you will see very big differences in how some of the signals are propagated. Well, when we think about signaling, of course, I like the little icons of the traffic lights. And that's sort of a metaphor for what's actually happening in signaling. And I'll tell you a little bit about what I'm going to talk about today. So I'll give you some background, obviously, which I'm doing right now. The considerations of membranes. Membranes for virtually all kinds of signaling are major considerations because they are, as we've talked about, impermeable to many molecules. And so if we're going to communicate information across a membrane, which is what's happening in signaling, then there are some tricks that cells have to use to do that, whether that is a nerve system cell or that's a liver cell that's responding to a hormone. Hormones and receptors are where I'm going to focus most of the attention on in both this lecture and the lecture on Friday. That's the subject of both of these lectures. And hormones and receptors are paired, obviously. Hormones are the signaling molecules that are extracellular, and the receptors are the things that interact with those hormones and communicate the signal. We will see that communicating that signal can be very complicated, surprisingly complicated. We'll also talk a little bit about second messengers, which are part of that signaling system. And second messengers are sometimes a misnomer because, as we will see, there are oftentimes other things we don't even call messengers that are communicating signals, and there may be dozens of those, proteins typically. Well, the needs of a multicellular organism, as I said, are quite specialized. Okay? Muscle cells have different needs than liver cells. Okay? An organism has to be able to respond to its environment. If it's not properly responding to its environment, you're in trouble. Okay? If you are needing food and your body is not signaling, for example, that it's hungry, then the likelihood that you're going to have consequences of not having enough energy are high. Well, most of us don't have too much trouble in recognizing or getting that signal that we're hungry. Some people do, actually. Okay? But most of us don't have that problem. The food status, all right, related to the same thing I just talked about. The food status being what have I had enough to eat? Do I have enough energy? Am I putting enough away as I need to function properly? Body has to be able to respond to danger. I often talk about the example of the grizzly bear chasing you. It's a very visual image of being out somewhere and someone or something is after you. Being able to mobilize your resources as best you can to respond to that danger is really critical. All right? The body has to grow. So you might not think about it, but the body, again, has to make decisions. Is it time to stimulate growth of bone cells? Is it time to stimulate growth of nerve cells, etc.? All right? And that growth has to be managed. My 2 legs are reasonably the same length. And that's because that growth was managed, and we have growth hormones that help cells to make that decision about whether or not to divide. And the body also has to respond to injury. We've talked about blood clotting and the healing process a little bit. And not surprisingly, the body has to grow new tissues. The body has to do new things, make new bones, for example, if you break a bone, to be able to respond to injury. Well, the metaphor of all the cells in the body pulling their oars at the same time in a coordinated fashion is a good one, all right? Because if, for example, one part of my arm muscle is deciding, okay, I am burning ATP, I need to contract, and the other part of that same muscle is working counter to that, then I'm not going to have a very good contraction. I'm not going to function properly. So it's important that that information be communicated in a consistent fashion that is, again, in the interest of the organism. I want you to keep that in the base of your thoughts, right? In the interest of the organism. When we look at signaling, signaling happens through the general process that we see here. First of all, we have a signal. And we'll talk about different kinds of signals. And I will also tell you that while I will include the nervous system in part of this signaling process, this set of signaling lectures is not focused on the nervous system. So the nervous system I will consider separately in other lectures, and that will actually happen next term. But I will remind you that signaling via the nervous system is a kind of signaling, but it's a very different kind of a signaling than what I'm talking about today. Most of what I'll be talking about today is signaling that's happening through signaling molecules in your bloodstream. Anybody with me on that? Nobody is with me on that? This is the quietest class I've ever had. What is this? Everybody's nervously looking away. Don't look at me. Why is that? Why are you guys so quiet? It's what? It's difficult? Am I scary? I hope I'm not too scary. But I think it's important. You know, I like questions, ask questions. I like interaction, and I just feel like I'm not connecting there somehow, so I'm not quite sure what we need to do there. It's the room? Does the room inhibit that? I happen to like the room. I've been very fond of it. In fact, I keep bringing people over to show them this room because I like it. But as a student, do you like the room or not like the room? No. How many like the room? How many don't like the room? Interesting, interesting. What do you not like about the room? I'm just curious. Too much PowerPoints. 8 screens is too much? Yeah. 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 The process. You're not getting enough time to process. You know, I'm very happy that you said that. As you know, I hate PowerPoint, okay? I used to teach, and I've been actually thinking about moving back to this because I'm wondering if this actually is a factor in the interaction component, that I used to lecture from an outline that was HTML linked. So I could click back and forth as I chose, and the student could see the bigger picture of the outline. So you see the forest, and I think what you see in PowerPoints is you see the trees. And I think what you said is very true. I'm lost. I'm looking at one slide, and where in the hell are we? So yeah. Yeah, so I'm describing it with my mouth instead of going through and doing that. Okay. Okay. Okay, great. Other feedback. I really appreciate that. Yeah. You see everybody. Yeah. The room is a little overwhelming. For me, that's good. Okay, I like to be overwhelming, I guess. But I feel like the person that's sitting in the back row, I can see them in their eyes, which I like, which I couldn't do in Milam Auditorium. And some of you probably have had classes in Milam Auditorium. If you're up in the balcony, you're so far away that I feel like I can't reach people. So from my perspective, that's good. But from your perspective, sitting and staring at that person across the way may not be the best, right? Okay, that's interesting. Yeah. I know. The acoustics in this room are really odd. When I've brought visitors to show in this room, I'll go sit in the back row and I'll say, sit up here, and I'll just talk like this, and it just, everywhere it goes out, you know? It's interesting. So I guess you're a little intimidated about talking? Is that right? Distracting, okay. Is the front row better than the back row? Okay. Shh, don't tell anybody in the back row that, right? What else? Yeah. Okay. I hope I'm not missing questions. So if you have questions, please don't be afraid to ask questions. I really like to interact, and I like to interact with you. And I'm feeling, a difference, and I'm certainly feeling a difference with the PowerPoints. And I'm thinking it's probably not going to be something I can change this term, but probably next term I'm thinking seriously about moving back to what I was doing. The reason I moved to doing PowerPoints was because this thing, if I do HTML, I always have to come back to this and click and go do something, and then come back to this and click and do something. Yeah, people carry around iPads and people carry around these wireless mice, but neither of them are consistent, and they're annoying if they're not working, in which case you always then end up coming back to this. But I think coming back to this may be what I'm going to go to, so I may do that next term. Yes? Yes. So what if I did a demo? That's a very good question. My wife asked me the same question last night. And I said, well, okay, given the fact that I'm just scrambling to stay on top of PowerPoints right now, as you guys are seeing, that kind of adds to it. I will try and see what I can put together for you, okay? It probably won't happen, it definitely won't happen before next week. I'm not sure if it'll happen next week, but if I can do that, I will, okay? I really do want to take your suggestions. Yes, back down here. Well, back here first and then down here. Yeah. Yep. Yeah. Yeah, that's what my wife said also. I should talk to you guys first, right? So she said, well, why don't you try that? Why don't you put an outline on one side and the PowerPoint on the other side? And I said, well, A, of course, I'd have to create it, which I'm happy to do. But B, I'm not sure that it helps with that navigation because I'm still having to click, 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 click through things to get there. And I would rather, if I'm going to make the outline, I would rather just make it hyperlinked so that I'm at that point in the first place. But it's a reasonable suggestion. It is. Yeah. That was your suggestion as well? Okay. All the minds are thinking the same thing. Yes? Yeah. Okay. Okay. If I go to make the outlines, I probably will just navigate to that. That's my thought, because it's a fair amount of effort to put these together from scratch, and if I'm going to put something together from scratch, I'm inclined to just do the HTML in the first place, which is why I'm thinking it'd probably be next term. So I will probably do that. But thank you. I do appreciate the feedback. Yeah. I want you guys interacting. Yeah. Back there. Okay. Okay. Concept maps. Okay. If I'm going through things and things are, whatever, dragging, or you have questions or you have a suggestion, just stop me and say, hey, let's look at this. This is clunky, right? I'll probably agree with you. That was what I was talking to my wife about this last night. I'm sitting there thinking, I don't want to be one of those boring professors that just clank, 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 which is all that's really happening, and people just sort of read their slides. And I'm finding that's what's increasingly happening as I'm going through these. So I really appreciate the feedback. Yeah? How much is he trying to get through? Uh-huh. Yeah. Right. So you're finding it scattered because, and you don't see the connections, which is what happens when you're doing a PowerPoint, because you don't see that bigger picture. That's exactly what I'm talking about. And I think you're right. I very much think you're right. Because where do you connect from one slide to the next? But if you see the outline and you say, I was over here and then I moved down over to here, and then I move back and I move over to there, right? Or somebody asks a question, I can go back, then that makes that so much better. So, yeah. Okay. Thank you. Thank you very much. It's always fun how the questions just seem to go opposite sides. Yes, ma'am. Yeah. I tend to go through things fast, and that's true whether I do HTML or I do that on PowerPoint, but I understand that, which is one of the reasons I am happy to have the videos available for you. And I'm also happy to slow down. If I'm going too fast for you, you can also slow me down. I don't have a problem with that. Yeah. Uh-huh. Okay. Yeah, makes you think harder about them. I like you being more involved in that, but I also want to make sure that that involvement is not at the risk of you having to be so involved that you don't understand in the first place. It's hard to be more involved. So there's a balance that has to happen with that. I certainly understand that. Yeah. Okay. I hear a voice. Oh, yes. Uh-huh. Yeah. Okay. Instead of just telling you. Okay. All right. That will take a little adjustment for me, but yeah. Okay. I appreciate that. Fortunately, we don't do too many mechanisms, but I do hear what you're saying. Mechanisms are kind of a pain, aren't they? Yes. Yeah, putting in the highlights, basically an overall design helps. That's a little hard to do, but I understand what you're saying. I would prefer to have it from the starting point, which I think makes a little bit more sense, but I agree that could be helpful. The information coming out isn't necessarily hierarchical, but the organization about how that you got there, I think, is. And that's why I think it's easier on the design than it is on the output. Because on the output, you go, where did this come from? Right? Okay, well, I'm going to have to move ahead, but thank you very much for that. I will make some changes, and I do appreciate your feedback. And as I say, if I'm going too fast or something like that, then please do let me know, okay? Talk fast. All right. Well, as I was saying, as I was asking the question or commenting, signaling happens largely by the process that we see on the screen. We see that a signal comes out. That signal, for our purposes, we're going to be talking about is largely hormonal in nature. We're not going to be talking about nerve cells. And that signal then interacts with cellular receptors. Okay? Those are the one or more intermediary molecules. There's one receptor that it will interact with, and that receptor will then interact with other things that communicates the signal. That intermediary molecule then causes changes to happen within the cell. And I'm going to go through, I'm not going to talk about those here, but you'll see that it causes changes in how metabolism occurs in some cases. It's going to cause changes in how gene expression occurs in other cases. And those are two biggies in terms of ultimate actions of hormones. As I said, the lipid bilayer is a barrier. We've heard this numerous times, and I've shown you this figure before. The lipid bilayer looks like that. We'll talk more about the lipid bilayer next term. And it's a barrier because it's got both nonpolar and polar components. And because of that, most things don't move across the lipid bilayer very easily. We will see one exception to that are steroid hormones. They actually move across reasonably well, which allows those types of hormones easier access into the cell. The lipid bilayer, of course, isn't flat as you see on the top, but rather is arranged in a sort of I don't want to say spherical, but a three-dimensional shape like you see below to form the outlines of the cell. Okay? So for information to move across the membrane, we give it a term. The term that we give it is signal transduction. Okay? Transducing something is to move it. So signal transduction is we're moving a signal, and we're moving it across the membrane. If the signal doesn't make it across the membrane, then the cell has no way of knowing what's going on. The cell is totally dependent upon those signals for it to understand what the body is doing, what the body needs. Okay. Now, one type of signaling I'll talk about here that relates to nerves, actually, at least indirectly, is the simplest type of signaling. The simplest type of signaling is what's known as ligand-gated ion channels. We're looking at a membrane of a cell, and we see on the left, okay, this receptor that's there in the blue embedded in the lipid bilayer. The signal is coming by this thing called a ligand, and the ligand is a small molecule, or in some cases, it's a protein. It can be either, depending upon the nature of the signal. The signal will be paired with a specific receptor. So this is a receptor that's specific for that ligand that you see on the screen. And the binding of that ligand, the hormone, ligand equals hormone, the binding of that hormone causes a change in the receptor. And the receptor here is something that is what we call a gate. And the gate is an opening that allows specific things in. In this case, the specific thing allowed in is calcium ions. This is an instantaneous thing. When the ligand binds, the gate opens. Things move across the gate, in this case, calcium ions selectively. Okay? And we see an ion change across the membrane. We see a voltage change across the membrane because we have charges that are moving. Right? And this causes the cell to adapt in some way. This could be a nerve cell, for example. And if this is a nerve cell, we've initiated a signal that's going to travel along that whole nerve cell and then communicate ultimately back to the spinal cord or the brain. Okay? So this is a very simple type of signaling. Okay? Hormone does one thing, and the response is that in this case we get an ion moving in. Okay? There's our response. Nerve system signaling, as I said, I'm not really going to talk about here. That'll be something I'll talk about next term. But we know that we're full of nerves. Some people have more nerve than others. And nerves are aimed at helping the body to respond. Sticking my hand in the flame, for example. My body says, get it the hell out of there. Yeah? Yeah. Is the receptor... No, the receptor is a protein. So the receptor is the purple thing in the middle there. That's what the receptor is. So we see... Oh, I didn't label the receptor. Good question. So the receptor is the protein in the middle. Okay? Yep. So that I should have labeled that. That's what the receptor is. But the signal is the ligand. So whenever we communicate with signal transduction, we have a signal and we have a receptor. Always. But good question. Yeah? What causes your body to recreate the ligand? If a grizzly bear is chasing me, my body makes adrenaline. My adrenal glands start dumping it out, saying, this guy's in trouble, get him the hell out of here. So we have largely glandular tissue that will make many of the hormones that we need to respond. There are other cell-to-cell communications that can happen as well. Okay? Okay. So nerve system signaling, as I say, I'm not going to talk about here. We will talk about that next term. All right, so we're going to focus on hormones signaling, which, as I say, occurs largely through the bloodstream. Hormones are the signal. Hormones communicate messages. That message can be divide. That message can be run. That message can be make glucose. That message can be break down glycogen. It can be a variety of messages that are being communicated by a hormone. Hormones are made in one part of the body, to answer the question that was just asked, typically glandular tissues for things like adrenaline. Okay? They're made in one part of the body, they exert their effects in the other part of the body, and they're traveling via the bloodstream. So they can only travel as fast as the bloodstream moves. And that's actually reasonably fast, on the order of seconds. The hormones, which are the signal, we call first messengers. First messengers. So the first messengers are the things that are moving through the body to communicate the information. They come in a variety of chemical forms. I've got 4 of them here. You don't need to memorize the structures. Okay? There's a thyroid hormone. Thyroid hormone is the only molecules in your body that are bound to iodine. That's why you have to have iodine in your diet. Okay? You see those I's on there. Those are iodine molecules. Epinephrine is also known as adrenaline. Several of the hormones that are important in our body are made from tyrosine. If you remember the structure of tyrosine, epinephrine looks a little bit like tyrosine, and they actually are derived. Epinephrine is derived from tyrosine. Some hormones in the body are proteins. Epidermal growth factor is a protein that tells cells it's time to divide. Growth factors will do that. Okay? And some hormones are steroid hormones. We call them steroids because they're derived from cholesterol. And when we study the structure of cholesterol later, we'll see it looks very much like what progesterone there is, and progesterone is ultimately derived from cholesterol. Okay. Cellular signaling has some complexity. I'm going to convince you of that in just a second. And the responses of signaling, as I said, are to respond to and help benefit the organism. You want complexity, folks? There's complexity. You want to go through all that for the next exam? No, I get a response on that one, right? We're not going to do that, all right? And I just simply show you this to remind you that what's happening as a result of a single signal. We could look at any of those things outside the cell. It could be one molecule that's doing something, and you look at the cascade of events that's happening inside, and you think, wow, why is it so complicated? Why does it go, all right, from this to this to this to this to this? And we're going to see a little bit of that later today. Why does it go through so many things? And I'll talk about that a little bit more next time, but part of the answer is it gives the cell flexibility because each of those can branch. More branches allow for more possible different directions for a signal to be communicated. Okay? More branches give more possibilities in terms of the signal that can be communicated. Now, we're not going to go anywhere near the level of depth that we see here. I am going to focus on 2 or 3 signaling types in cells, and I'm going to lead you through those, one of which I'll go through today. There are, for the purpose of hormone signaling, 2 types that I want to talk about. One is steroid hormone signaling. Steroid hormone signaling is in a class by itself because steroid hormones can cross the cell membrane. They cross the lipid bilayer reasonably easily. You can see that happening here. And so when the steroid hormone interacts with the receptor for the steroid hormone is inside of the cell. It's not membrane bound. You can see it floating around there in the cytoplasm. Some receptors float around in the nucleus. Okay? So the interaction between the hormone and the receptor happens after the steroid hormone gets into the cell. That's only true for steroid hormones. It's not true for other hormones. Other hormones, okay, there we have the receptor. Actually, maybe before I talk about other hormones, let me say one word about steroid hormones. Steroid hormones tend to have the simplest signaling pathways. You saw the complexity of the last slide? Look at the simplicity here. It comes in, it binds to a receptor, and the receptor goes down to the DNA and affects how RNA is made. Very simple. Connect, affect transcription. That's really what happens with steroid hormones. The other hormone signaling that we'll talk about has more complexity, and it can go a variety of directions. We can see in this case that we have a non-steroid hormone at the top. It is bound to the receptor, and the receptor is embedded in the membrane. That means that there has to be something that happens to that receptor that allows the inside of the cell to know that a hormone has bound on the outside of the cell. That signal transduction process is different for a non-steroid hormone. Steroid hormone makes it in, the signal goes. Non-steroid hormone binds to a receptor. That receptor on the inside has got to tell the cell, hey, I bound to a hormone. Okay? That's a little bit more complicated of a process. And it's more complicated than we see. That signal goes from one protein to the next protein to the next protein to the next protein, and whoa, that's a lot of information. Surprisingly, that travels pretty quickly. We'll focus on a couple, as I said. Now, that ultimate response of the non-steroid hormone can be transcription, and you can see at the very bottom, this particular one is leading to transcription. It might also be affecting the activity of enzymes to affect metabolism. And that's the example that we'll see today. Okay? Well, binding, if we're talking right now about the receptors that are in the membrane, which are the ones I'm going to talk about first, we see that the binding of the hormone has got to cause some change. So if we look at this receptor, we see that after it's bound to the hormone, something on the inside, which is the bottom of this receptor, the shape has changed. So again, small changes in binding of things cause other small changes in the shape of a protein. And these small changes are actually the signal that something has bound. Okay? So the one on the right, on the inside of the cell will know it is bound to a hormone because the inside of that receptor's shape has changed. Okay. The first type of signaling I want to talk about are what are called G-protein coupled receptors, or you can call them GPCRs. This is an example of a GPCR. It is a non-steroid binding receptor. It's bound in the lipid bilayer, as you can see here. And this is another view of the same thing. The lipid bilayer here is shown in gray. And this particular receptor is a class of receptors called 7TMs. M is in Mike. Okay? They're called 7TMs because TM stands for transmembrane. And you can look in this guy, and you can see in blue, and light blue, and green, and in red a little bit, though the red doesn't show up very well here, that this thing goes up, down, up, down, up, down. It crosses back and forth across the lipid bilayer 7 times. That's where the 7TM comes from. Okay? So 7TMs are very common types of receptors that we see inside of cells. And the first ones that I'll talk about are actually 7TMs. There are almost 800 genes in the human genome, okay, that are of this type. And interestingly, 460 of them are in our nose. Now that may sound like a lot, but if you look at the number of receptors that a rat has in its nose, it's about 4 times that of humans. Okay? Our nasal receptors aren't nearly as good as many other organisms. The example I'm going to talk about is called the beta-adrenergic receptor. Adrenergic referring to adrenal, and adrenal referring to adrenal glands. So the adrenaline that I talked about, these are things that are going to bind adrenaline, or as it's otherwise known, epinephrine. Okay. So it's membrane bound, 7 transmembrane domains. Extracellular on the top, intracellular, in this case, on the bottom. So we have an outside part and we have an inside part. Okay, how does a GPCR work? Well, we start out, first of all, with a hormone. The body has released a hormone, it travels in the blood, it gets to its receptor, and the receptor binds to it. Now, receptors have to be specific for specific hormones, right? Just like an enzyme has to be specific for specific substrates, a receptor has to be specific for a specific hormone. We don't want every receptor responding to every hormone because that wouldn't make any sense. Okay? The binding of the hormone causes a structural change, as I've talked about. And that structural change, okay, replaces, in this case, actually, let me back up. The structural change that's here affects the receptor's binding of an internal protein. So you see the internal protein in pink on the bottom right. What does that do? Well, this particular protein is called a G protein. That's where the GPCR comes from, the G. Okay? A G protein does a lot of things. We'll talk a little bit more about G proteins later, but the G protein binds to guanine nucleotides. That's where the G comes from in its name. The G protein is interacting with the adrenergic receptor. Yes? Yes. So the question was, did I say the receptor changes and the protein changes? The answer is both happen. So the binding of the hormone causes the receptor to change. The receptor's interaction with that G protein changes. And so the G protein itself changes. It was kind of like when we talked about hemoglobin, binding of one oxygen caused the protein to change, and that affected the interactions with the other ones, which caused them to change. Same principle going on here, only now we're talking about a receptor and a G protein. Now, this change in the G protein is critical to the communication of the signal. In fact, every step in the signaling process is critical. When we screw up signaling, we frequently end up with cancer. That's not good. Signaling, okay, is one way that cells have of controlling their division. If we screw up that control, we might realize that we have uncontrolled division, and uncontrolled division is cancer. So signaling is really, really critical to the proper functioning of a cell in the body. Every step is critical. Okay. So that change in the G protein here causes the G protein to let go of the GDP that it had. If you see on the top panel at number 3, you see that G protein was bound to a little blue ball, and the binding of the hormone caused it to grab the little yellow ball that's labeled GTP. So it's letting go of GDP, it's picking up GTP. Well, you could imagine if I change what the protein is bound to, the protein itself is going to change in some way, right? So the G protein changes, and when the G protein changes, you see in this case that one of the subunits breaks apart from the other two. This subunit is called alpha. Alpha is the part that binds to the GTP. When alpha is bound to GTP, it lets go of the other subunits and goes off and does its thing. Okay? That going off and doing its thing can cause an enzyme to be activated. This is where an outline would be helpful, wouldn't it, guys? I'll draw you an outline. How's that? Okay? I'll make you an outline for this. It's going to go and it's going to activate an enzyme. And the other thing that alpha subunit can do is interesting and odd. It won't seem important at the moment, but it will later, and that is it can break GTP down into GDP. It turns out this subunit can turn itself off. Binding GTP turns it on. Breaking down GTP turns it off. So it turns itself off. After it's done that, it can return to the original state that it was in, and the whole cycle can continue again. Yes? What's that? What happens to the hormone? Very good question. The hormone can be released by the receptor because it's not covalently bound. And if it doesn't get released, the cell has a kind of an insurance policy to turn off the receptor. And I'll talk about that next time. But that fate of the hormone is important. Yep. Okay. Membrane receptor proteins internalize the message. You've seen that. Activates the synthesis of second messengers. I haven't talked about that yet. Okay. Here are some second messengers. What second messengers are the next part of the signal. You've seen the first part of the signal. You've seen the signal getting in. You've seen the G protein get activated. And when I said the G protein is going to go and activate enzymes, those enzymes make second messengers. Second messengers have a variety of forms. The one we're going to focus on is cyclic AMP. These are all second messengers that are on the screen. Cyclic GMP is commonly used in vision. Okay? No, you don't need to know the structures. Okay? What these second messengers do is they play a role in the communication of the message. And one of the things that they stimulate is the covalent modification of proteins in the cell. You remember that thing I showed you where I had a whole string of them, one to the next, to the next, to the next, to the next? Those are all covalent modifications, like we saw when I talked about the blood clotting. I couldn't pull it out. Blood clotting, right? Okay. The result may be altering gene expression. That's the transcription I've talked about. The result may be changing an enzyme activity affecting metabolism. Okay. Yeah, question? Yes. So are the second messengers, what she said, activated by the first messengers? The second messengers are created in response to the binding of the first messenger. So the answer is yes. Yeah. I wouldn't say they're activated by that, but they're actually created as a result of the binding of the first messenger. Okay? Yeah. So the first messenger, I'm going to sort of summarize his question with my answer. The first messenger binds to the receptor, which causes a change inside that results in activation of an enzyme that creates the second messenger. Okay? And I'm going to give you an example here that I think will hopefully help clarify that. Okay? Let's look at how a GPCR works. Here's a GPCR. That's the membrane receptor. We can see it's bound to the alpha subunit here that is labeled GDP. The GDP is part of the alpha subunit. There's also a beta and a gamma subunit. That's the other two parts of that protein. It's in the resting state. Nothing has happened. Okay? Heterotrimeric, meaning there's three different proteins, an alpha, a beta, and a gamma, that make up a G protein. The G protein associates with the G protein coupled receptor. That is, they associate, as you can see here, and as you saw in the previous figure. The G proteins get altered by the binding of the hormone. Again, this is what I've told you already. In this case, we're looking at one that binds to adrenaline, epinephrine. The release of epinephrine into the bloodstream causes the G protein coupled receptor to change. We see that change going from a yellow square to a green pentagon when it's bound to the hormone. And that causes the G protein to be altered. We see here that the G protein is letting go of the beta and the gamma. The alpha is letting go of the beta and the gamma. And we see replacement of the GDP with GTP by the alpha. Now we've got an active alpha. Again, I've shown you all this before. I'm just showing you in a different figure now. The alpha is released, and that alpha can go do something. Now I'm going to show you what that alpha goes and does. So the GTP bound to the alpha means that the alpha is active. GDP bound to alpha does not make alpha active. Okay. So the second messenger is going to be created next. Here's the active alpha subunit bound to GTP. In this case, the alpha subunit goes and binds to an enzyme in the membrane of the cell called adenylate cyclase. This is a very common route for G proteins. It binds to adenylate cyclase in the membrane, and when it does, it changes the adenylate cyclase. And you'll notice I've changed it from red, meaning off, to green, being on. Adenylate cyclase is activated by binding of an active alpha subunit of a G protein. Okay? When it's active, this enzyme catalyzes the conversion of ATP into cyclic AMP, and you might remember that was one of the second messengers that I talked about. So now we've seen how this message has been communicated. It's gone through a couple of phases, and we've seen the second messenger be created. The second messenger is now going to travel through the cell and communicate that information. There's the second messenger. How does that happen? Well, we're going to follow one example here. Question? It's kind of like a cascade. It is. We'll see more of the cascade coming up. Okay? Yeah, question? Up. When what travels? Cyclic AMP? So the alpha subunit is pretty much close to the membrane because that's where all of its binding things are going to be. It's not in the membrane, but it's close to it. Okay? The cyclic AMP can go anywhere. All right. Here's the cyclic AMP. I've labeled 4 of them, and I've made them smaller so I can fit them on the screen. 4 cyclic AMPs interact with a protein called protein kinase A. And yeah, there's a lot of steps to this. Okay? We're only about halfway through. There's 4 steps to this. All right? Protein kinase A is the next messenger in the process. It doesn't give it a name as a messenger, but it is a messenger. We don't call it a third or anything like that. Only the small molecules do we call second messengers. Protein kinase A, in the form that you see on the screen, is inactive. It has 2 R's, which are regulatory subunits, bound to 2 C's, which are catalytic subunits. And in this state, the regulatory subunits are covering up the active site. When it binds to cyclic AMP, the regulatory subunits let go of the catalytic subunits, and the catalytic subunits now, with the little dazzling lines outside them, are active. They can do what these catalytic subunits do, which is to put phosphates onto proteins. That's why it's called a protein kinase. A kinase is something that puts a phosphate onto something. So this kinase is putting phosphates onto proteins. It's specifically putting phosphates onto serine and threonine side chains, because they're not. And now we've made a protein that didn't have a phosphate into one that has a phosphate. Do you suppose we would change that protein's activity? You betcha. So we see the information is passing along. This message is being communicated through. Now we've got a phosphoprotein. Okay? All right. Now, I'm not going to have time today to finish what I'm going to start showing you. How much time do I have? I've got maybe 1 minute to show you something. All right? So I'll come back and start here next time. Yes, we have a song, but I want to start this so you can maybe have some context for the song. Here's what I want to show you. You can see some of the things at the top of the screen that I've talked about before. And this is how our body controls the breakdown of glycogen. Glycogen is a polymer of glucose, as you may recall. Breaking down glycogen is a way for the body to release glucose into the bloodstream. So if I release epinephrine, the body gets a giant release of glucose into the bloodstream, and consequently the muscles now have a lot of energy to go do things, and that's why when people get adrenaline, you hear stories about them being able to lift up a car because of danger or something that they perceive to someone. That happens because of the dump of glucose that happens from this pathway. Now the song I'm going to play for you is going to incorporate some of this, so some of it we're going to cover next time, but I think you'll enjoy this song. Let me get to that song. And then I'll come back and talk more about this next time. Thank you, by the way, before I finish, for the feedback that you've given me. And we shall. It's called the Tao of Hormones. Time to study you again. Mechanisms that I need to know. Things that really stress me so. Pathways planted firmly in your head. Ahern said with epinephrine. And proteins are well known. Changed unbinding this hormone. Rearranging cells without protest. Stimulating Ig-alpha S. To go open up and splice this GTP with GTP because of epinephrine. Active G then moves away, simulating at cyclase. So a bunch of cyclic AMP binds to kinases and sets it free. All the active sites of the kinases await triphosphate because of epinephrine. epinephrine. Muscles are affected then, breaking down their glycogen, so they get a lot of energy in the form of lots of G1P. The synthases that could make a glucose chain all refrain because of epinephrine. Now I've reached the pathway end, going from adrenaline. Here's the trick I learned to get it right, linking memory to flight or fright. So the mechanism that's the source of anxious fears reappears when I make epinephrine. Okay,